Welcome back to the Answer Series Diagnostic Report teaching and learning tool for the NSC November examination, an all-in-one tool that combines the paper, memorandum, and annual diagnostic report commentary to create a comprehensive teaching and learning experience. TAS provides this document for free, and we encourage teachers, learners, tutors, and subject advisors to use it. All we ask is that you please give credit where credit is due and not change or alter any content in this document that removes our name from it. Continuing with question 3, the passage describes the breeding habits of vultures. Vultures are birds of prey that feed on the flesh of dead animals. A female vulture lays only one of 2 fertilized eggs, which are then protected by both parents during incubation. Incubation is the period from the time an egg is laid up to the time it hatches. Vultures have a short incubation period of 32 to 45 days. This short incubation period is typical of birds displaying altricial development, and after hatching, the parent feed the young chicks as often as 20 times a day. The first question asks us to give one reason from the passage why vultures are considered as oviparous. So learners had to remember that oviparous means that it is egg laying, and that's also mentioned in the second sentence. A female vulture lays only one of 2 fertilized eggs. The diagnostic report mentions that candidates did not read the instruction to quote from the passage and instead gave a definition of oviparous. The second question there asks for one reason from the passage why vultures have a high survival rate, even though few eggs are laid. So the survival rate here refers to the fact that they can actually grow up to be big vultures, and there are a few instances in this paragraph which talks about the fact that the parents actually look after their young very well. The first is that it says that the eggs are protected by both parents during incubation. And then it also mentions that the parents feed the young chicks as often as 20 times a day. So they're constantly there to feed the chicks and to look after them. 3.1.2, fertilization in vultures takes place internally. State 2, advantages of internal fertilization. So internal fertilization is advantageous because it increases the chances of fertilization. The gametes, the sperm and the ovum, are quite close to one another as compared to with external fertilization, where these are released into the environment at large. Gametes are also protected from predation or from drying out as desiccation, or any other environmental factors like, for example, rivers or streams actually just washing these gametes away from one another, which is often the case in external fertilization. Water is not needed, it's not a requirement, and fewer gametes are needed, so that's also an advantage because it means that the body has to expend less energy in the production of gametes, as fewer of them are required, and there's a high chance of them actually meeting up and causing fertilization. Any 2 of those would have been correct. The diagnostic report says that candidates referred to the advantages of internal fertilization for the embryo or the fetus, in other words, how that protects the developing embryo, instead of how it's advantages for the gametes. So let's revise this. So just a basic understanding of the difference between internal and external fertilization is provided. Here you can see a picture from the 2018 NSC March paper where we have a representation of external fertilization taking place. The male releases the sperm into the external environment. The female releases her ova into the external environment, and the hope is that these 2 gametes will meet up at some point to create embryos. So mostly fish and amphibians perform external fertilization. On the other side, we do have an example of amphibians that perform internal fertilization, the 2 frogs there from the 2023 NSC November Paper 1. The male and the female are copulating, which means that sperm are transferred directly into the female body via the male copulatory organ, in other words, a penis, for example. So internal fertilization means that it occurs inside the female body, so the gametes are protected by the female body and they are in close proximity as well, which increases the chances for fertilization. Mostly insects and terrestrial or land vertebrates, for example, reptiles, birds, and mammals, exhibit internal fertilization. If we continue learning, we have a nice summary table of the differences between internal and external fertilization, their pros and cons to some extent, as extracted from the Answer Series Grade 12 Life Sciences Part 1 class text and study guide. So in terms of external fertilization, the first problem or con of external fertilization compared to internal fertilization is that basically gametes are exposed to the environmental elements when they're released outside the body, for example, air, and that can dry them out. A potential solution to this problem is that animals that perform external fertilization will often do so within aquatic or moist environments. Think of, for example, the fish in the previous slide that release their gametes into the water. Why is this important? Because it prevents the dehydration of the gametes and or embryos. However, internal fertilization has a benefit over external fertilization in this regard, which is that the gametes are not released outside at all. Internal fertilization ensures that the gametes are released inside the female body and therefore they're not even at risk of dehydration. When we're considering the embryo, it develops inside the female body, which is also not at risk of dehydration, and reproduction is therefore also not limited to aquatic environments. So animals that are land living tend to perform your internal fertilization because they don't require the water environment. The second problem with external fertilization is that gametes are easily lost because of predation or maybe damage from the environmental factors. A solution to this is that animals that perform external fertilization will often produce a large number of gametes. And the reason for this is that at least, even if some of them do not survive, there will be enough of them to ensure that some of them are fertilized. So it increases the chances of fertilization given the disadvantage of the potential damage to gametes. On the other hand, internal fertilization overcomes this because gametes are released inside the female body and therefore they're not at risk of predation or exposure to damaging environmental factors at all. Embryos develop inside the female body and they're also not at risk of predation or exposure to environmental factors that might damage them. The third problem with external fertilization is that embryos or offspring are easily lost due to predation or damaging environmental factors. So in other words, once the gametes have been fertilized and the embryos stay outside, the body may be connected to some kelp or algae, and they're sitting there just developing into little fish or octopus or whatever the case may be. A large number of these can ultimately be eaten by other fish or washed away. So predation or damage creates a problem for offspring, the embryos themselves. The solution is again that because there is a large number of gametes being produced, you have an increased chance of fertilization, and obviously that leads to a large number of offspring being produced, so that it ensures that at least some of them will survive until adulthood. So some of them might be eaten by other fish, but at least there are enough of them that maybe 5 or 6 survive. Internal fertilization overcomes this problem because fewer gametes are required, because there's a greater chance of fertilization happening inside the female body. The gametes are in closer proximity in an isolated system. Inside the female body, gametes are also not at risk of predation or being exposed to damaging environmental factors. And then the embryos develop inside the female body where they receive the protection and the nutrition that they need from either a yolk in the case of an egg or a placenta in the case of a viviparous organism. Then another problem with external fertilization is that sperm and ova are released separately into the external environment, which means that they need to find one another, which is a bit of a problem. So the solution that animals have for this problem is that courtship and mating behavior leads to the male and female coming in close proximity, or maybe they release pheromones of some kind to attract each other. This ensures that the sperm and the ova are released at the same time, or at least in the same space, to increase the chances of fertilization. Internal fertilization overcomes this because the sperm and ova are always going to be released inside the female body in close proximity and often simultaneously due to copulation events. This ensures a higher rate of successful fertilization. And then lastly, external fertilization requires a lot of energy because it produces a large number of gametes, and very often a lot of those gametes don't survive. So it's a lot of energy to put into something that has a very low rate of success compared to internal fertilization. But it produces such a large number of gametes to try and increase that success rate. So the solution is that the developing embryo or offspring are independent. They require very little parental care. So at least we're offsetting that energy input that initially happens in the production of the gametes. We offset by not having to require too much parental care after fertilization and development. And very little cooperative behavior is required between male and females. So for fish, it's just literally releasing sperm and ova into the environment and hoping they get enough contact between them. And the explanation for this is that obviously it conserves energy of parents during that stage of their development. But where internal fertilization has the advantage here is that less energy is going to be spent on the production of gametes because far fewer gametes are produced in internal fertilization. But it does have a disadvantage compared to external fertilization, which is that Even though less energy is spent in the production of gametes, more energy will be spent in the parental care that's required afterwards. So again, that energy offset actually becomes a problem for internal fertilization. Also, mating strategies and cooperative behavior between males and females is required, which also says that, there's more energy required from their side in order to get the whole process going. Then if we continue home, so we continue on to question 3.1.3, still related to this little reading piece on vultures. It asks us to state 2 characteristics of chicks that display altricial development. And the diagnostic report says that candidates confused altricial development with precocial development. They don't really remember the difference between those 2 terms. So let's find a way to help learners to remember that. So here we have a little tip box that says remember these phrases and pictures to remember the difference between altricial and precocial. So when we think of an altricial animal, remember the phrase the adorable altricial ape, A, adorable altricial ape, because that helps you think of this little picture with a helpless baby ape that's clinging to its mother. Its mother has to carry it around, she has to feed it, she has to keep it warm, and she has to protect it from other predators. So an altricial organism is one that depends a lot on its parents after its initial birth. It's like us humans. Babies, we are very vulnerable. We're born with, not a lot of skills to survive the environment. So the adorable altricial ape creates this image of a newborn baby that requires a lot of help from the parents to survive in those first few months or maybe even years. Then on the other side of this is a precocial organism, pre meaning before. So it's actually ready before birth. It's rearing to go. So think of a newborn pony. So the phrase that you can remember is the prancing precocial pony. PPP, prancing precocial pony. So you think of this little frolicking, wobbly newborn pony just running around shortly after birth, covered in fur, eyes wide open, and just absolutely loving life. It can move on its own, it can regulate its own body temperature, it can run away from danger, it can come get some milk from mom, it doesn't have to sit on mom's lap and she has to feed it. Mom is grazing while it's drinking. So, it can kind of take care of itself. And that's the difference between altricial, like dependent, and precocial, ready before birth to some extent. En in Afrikaans kan jy dink aan die afhanklike altriciële aapie en die vrolike vroegselfstandige filletjie. Continuing back home, so getting back to the memo for question 313, the 2 characteristics of chicks that display altricial development. So think of the adorable altricial ape, so organisms that need a lot of care afterwards. Now if we apply this to birds, this means that they're probably born with eyes closed. They don't have any feathers or fur or down of any kind so that it covers their bodies and protects them against the cold. They're unable to move. Their parents have to move them around, or they have to stick to the nest because they can't find their own food, and their parents have to provide them with that food or provide them with protection from other organisms. Then question 314, explain why birds with altricial development would have short incubation periods. So these 2 things connect to one another, and it's important to look at the paragraph which says the incubation is the period from the time an egg is laid up to the time it hatches. So it's basically referring to the time when there is a development happening inside the little egg. That embryo is growing into a bird. Now, altricial development, we just said, means that the bird is born not very well or fully developed and has a high dependency on its parents to take care of it. Now, the short incubation period connects to that because it basically means that the egg couldn't keep it alive inside for very, very long. The only thing inside the egg that actually nourishes the developing embryo is the yolk. So the less yolk an egg has, the shorter the time for incubation will be, and the greater the chance of the organism being born, or in this case the bird being born, altricial, so that it's dependent on its parents. Now, the diagnostic report says that candidates found it very difficult to make the connection between the amount of yolk in the egg and the degree of development of the chick. So let's understand this concept once and for all. So let's look at birds as an example. Here we have 2 different species of birds. We have a chicken at the top and a budgie at the bottom. If you've never seen a little budgie, it's extremely small. when it hatches. Now note on the hand the difference in the size of the eggs. A chicken egg is much larger than a budgie egg. It has about 35% yolk compared to a budgie egg which has about 20% yolk. And a chicken egg hatches within 21 days compared to a budgie egg which hatches within 18 days. So you can see that the smaller egg has less yolk and it hatches sooner, because obviously the lower amount of yolk cannot keep the bird developing inside the egg for very long, compared to a greater degree of yolk in the chicken egg, which gives more nutrition to the developing chick, so it can actually stay inside the egg for longer and it hatches after 21 days. Now this is the difference between a precocial and an artificial species. Chickens are precocial birds, which means that they are born ready to go. If you've ever seen a little baby chick, you know that its eyes is open, it's cheeping all over the place. It already has its whole body covered in downy little feathers to keep it warm. Immediately it's active, looking around, walking around, and it can independently take care of itself for the most part. So a precocial species like a chicken has much less parental care that's required. And how this relates to its egg structure is that its egg contains more yolk, therefore the yolk provides more nutrition inside the little egg. The chick can stay inside it for longer. There's a longer period of incubation and therefore maximal development before hatching, which means the chick is rearing to go. It's ready to go. It's precocial. On the other hand, budgies are altricial species. So here you can see in the picture, if you compare the 10-day-old chicken or chick to the 10-day-old budgie, you can see a clear difference in the level of development. So altricial species like the budgie little chicks, they hatch not ready to go. Okay, they hatch with a lot more attention and care that's required. Their eyes are closed. They have naked bodies, no feathers to keep them warm. The parent needs to do that. They are not active in terms of finding food or hiding from predators. The parent needs to provide the food and protect them. They cannot walk and fly, so they remain in their nests for a long period of time. So in altricial species, we need a lot more parental care. So in terms of how this relates to their egg structure, their eggs typically have less yolk, which means that the egg can only provide so much nutrition. So the incubation period is much shorter and therefore minimal development happens before hatching. Most of that development has to happen after hatching, and that's where they're born, very dependent on their parents. Now, if you continue learning, we see a table that compares different species of precocial and altricial birds, the weight of the eggs that they produce, their albumin and yolk concentration. So the emphasis here is actually on the yolk and the species. So at the top we have our precocial birds like chicks. You can see other examples include ducks, turkeys and geese. They are all precocial birds, which means that their hatchlings are born rearing to go. They're ready, they're pre-developed for life. And what you can see is they can produce quite large eggs or small eggs. A goose can produce an egg up to 200 grams in weight, a chicken 58 grams in weight. The importance here is the yolk percentage. All of the yolks have a percentage greater than 30 in the precocial birds, which allows for a longer incubation period and therefore allows for more development inside the egg, which leads to them being precocially born. Artificial birds, on the other hand, like our buzzard, our pigeon, starling, golden eagle, and we now know budgies as well, you can see that their egg weights also vary quite immensely. You have a golden eagle just produces a very large egg, 140 grams, compared to a starling, which has a teeny tiny little egg of 7 grams. But again, the emphasis is not on the size of the egg necessarily, it's on the percentage of yolk inside the egg. And you can see that all the percentages for those altricial birds, the yolk percentage is far lower than 20. So that means that the egg can only provide so much nutrition. The incubation period therefore is shorter, and those birds are born not necessarily rearing to go. They have altricial development, which means they're dependent on their parents. So hopefully this kind of gives you a better idea of how to understand how the yolk percentage relates to the development of the organism and then how much parental care is required after birth. So let's go back home. So just again, looking at the memorandum for 314, explain why birds with artificial development would have short incubation periods. The chicks are not fully developed when they are hatched because the eggs have less yolk, or because there is a higher degree of parental care. So we understand now the connection between the yolk in the egg, the degree of incubation, and whether the organism then is born artificially or precocially. Question 3.2, the diagrams represent the pupillary mechanism in the human eye. You need to identify part X and Y. So part X is the pupil and part Y is the iris. You can see that that structure changes between diagram A and B, and the fact that the question talks about the pupillary mechanism tells you that these structures that are changing size and shape are the structures involved in the pupillary mechanism. Question 3.2, explain why the pupillary mechanism is considered to be a reflex action. Remember that a reflex action is a rapid and involuntary response, and the third mark is given to applying that definition to the example of the pupillary mechanism, which is that the rapid and involuntary response is going to be to a light stimulus in this particular instance. So candidates gave generic descriptions of reflex actions. Most of them would have gotten the first 2 marks, which is just basically the definition of a reflex action, but they lost the third mark because they didn't actually say that it is in response to a light stimulus. Question 3.2.3 asks you to name the 2 effector muscles that are involved in the pupillary mechanism. So in the pupillary mechanism, we have the iris, that is the structure that controls the size of the pupil, and it consists of 2 muscles, the radial muscles and the circular muscles. Question 3.2.4, explain the significance of the change in the diameter of part X from diagram A to diagram B. So they're basically asking you what happened. Why did this happen? How is that significant? What do you think caused that change to occur? So the first thing is that we need to state what we see. So from diagram A to diagram B, we see that part X became larger. Or you could say that the pupil, part X, dilated. Now, why did that happen? Most likely because this person was in dim light. That's the typical thing that happens when you are in darker light conditions. Why does it happen? So that more light can enter the eye and a person can actually see in the dim light. It improves the vision. So candidates gave both mechanisms for dim light and bright light, in the case of which a marker was instructed to mark only the first answer. So because candidates did not understand what the question was asking them, they just basically blurted out the whole pupillary mechanism. And if unfortunately they started with what happens in bright light circumstances, they would have lost those 4 marks, because this picture here from diagram A to diagram B is showing us what happens in dim light conditions. We know this because the pupil is dilating, it's enlarging. Then question 3.3 was one of the first scientific investigations. It says it was conducted to determine the effect of insulin on blood glucose levels. That is the aim of the investigation, and you always highlight that to ensure that you have it to work with. It says the procedure was as follows. 2 groups of 10 people each were selected. One of the groups consisted of healthy individuals, and the second group, which served as a control, consisted of people with diabetes. Each group was given 75 grams of glucose to ingest. Their blood was tested again after 60 minutes and then 90 minutes to determine the level of glucose and insulin. Note that the normal level of glucose in the blood is between 3.9 millimolars per liter and 5.6 millimolars per liter, and the normal level of insulin in the blood 60 minutes after the glucose was ingested is always between about 18 microunits per liter and 276 microunits per liter. And then they show you the table in which the results were recorded. If we look at the first question, it asks you to state the purpose of the control group in this investigation. So the introduction says that the aim was to determine the effect of insulin on the blood glucose levels. Also consider that the introduction stated that the control group consisted of a group of diabetics. So that means we have a group of people that don't have insulin in their body or struggle to produce insulin, And then we are comparing their results to a group of healthy individuals that do have the capacity to produce insulin. So what that's going to allow us to do is to see exactly what insulin does to the blood glucose levels. And so the control group in this case, consisting of diabetics, is going to ensure that any change in the blood glucose levels we do observe is as a result of insulin only, because that is the big factor that differs between these two groups. Now, the diagnostic report says that candidates gave generic answers here. So they just basically said the control group is there to observe the effect of the independent variable on the dependent variable or some other generic response. And they didn't link the function of a control group to this specific answer. So remember, in this scenario, we want to view the effect of insulin, and the control group doesn't have insulin or cannot produce insulin effectively. They're diabetics. So it allows us to clearly see that any change in the blood glucose levels that we do observe is due to the presence of the insulin in a person's body. Question 332 says describe 2 ways in which insulin decreases blood glucose levels. So you have to think of the insulin pathway. Insulin can decrease blood glucose levels by just increasing the absorption of it into the cells, and the body cells then use that to produce energy. It can also stimulate the liver to start converting glucose in the blood into stored glycogen. This can also happen in muscle cells, specifically your skeletal muscle cells, and it can increase the general cellular respiration rate in your whole body so that the glucose in the blood can be used up. The diagnostic report says that candidates couldn't describe how insulin decreased blood glucose levels. They described the mechanism of insulin production by the pancreas without linking it to the absorption and conversion of glucose by the cells or the liver or the muscles. Then question 333 asks us which group consists of healthy individuals, and that's the tricky part, because the question tells you the first group consisted of healthy individuals and the other group consisted of diabetics. But just because the groups are labeled X and Y doesn't mean they occurred in that sequence. So they want you to actually look at the data, understand the data, and then be able to deduce which group is the healthy individuals. And the second question, 334, then asks you to use the data in the table to explain your answer. So looking at the data in the table, let's start with what happens at 0 minutes. So if we compare group X and group Y before any glucose has been administered, we notice that the average level of glucose in the blood for group X was 6,8 compared to 4,5 for group Y. In the note, it tells you that a normal level of glucose is between 3,9 and 5,6. So if we look at that, it means that group X, the glucose level is above the normal range, whereas in group Y, the glucose level is within the normal range. Now, the note also tells us that we can look at the normal level of insulin in the blood 60 minutes after glucose ingestion. So now we have to focus on the 60-minute column. And we can just confirm our suspicion that group Y are the healthy individuals by seeing that a healthy normal level of insulin is between 18 to 276 microunits per liters 60 minutes after glucose ingestion. And if we look at the values of group X, we see that their average level of insulin is 2 after 60 minutes compared to group Y, which is 142. So again, group Y's value falls within the normal range and group X's value is very, very low. Outside of the normal range. And then we can also look at the 90-minute interval as a last possible place where you could have given a reason. At 90 minutes, if we look at the average level of glucose in the blood, we see that for group X, it stays quite high, 8,7, which is above the normal level of glucose in the blood. And for group Y, it has returned back to 4,5, its original value that is within the normal level range of glucose in the blood. So that's 3 different reasons. By looking at all the data on the table, we can find a reason for what happens before glucose ingestion, 60 minutes after, and 90 minutes after to confirm that group Y are the healthy individuals. So looking at the memorandum, obviously group Y then consists of the healthy individuals, and the data on the table we can use to explain this in 3 different ways. So the 3 reasons are that at 0 minutes before glucose ingestion, the blood levels for glucose in group Y was within the normal range of 3,9 to 5,6 millimolars per liter, whereas the blood glucose level for group X was higher than that. At 90 minutes after glucose ingestion, we can see that the blood glucose level for Group Y's participants has returned to normal within the normal range as 4.5 millimolars, whereas that of Group X, the participants there, the blood glucose level spiked and it has remained high at 90 minutes. And then the final reason is that at 60 minutes after the ingestion of glucose, the insulin level for Group X is very low and not within the normal range of 18 to 276 microunits. But for Group Y, the average insulin in the blood is within that normal range. It's 142 microunits per liter. So the diagnostic report says that candidates did not understand how to use the data to explain their answers. Many of them simply compared Group Y to the normal limits, and their answers were not specific. They failed to state the times at which the glucose was within the normal limits. So you had to basically combine what was given in the note to substantiate what was given in the table. That what we see in the data for Group Y makes them the healthy individuals because their data at those 3 different time intervals coincides with a normal level of glucose or a normal level of insulin. The flow diagram shows the homeostatic control of thyroxine in the human body, and let's first try and understand this very complicated diagram. So we can see that we have a gland Y and we have a pituitary gland. So let's start with the pituitary gland because I think it's just easier looking at something that has a name. So the pituitary gland, you're going to see there's less TSH being released, and that then targets gland Y. That changes the level of thyroxine in some way, X, which then again affects the pituitary gland to secrete more TSH. That affects gland Y once more to change the level of thyroxine Z. So something happens to the levels, and then this goes in this kind of circular motion the whole time. So that's typical of a homeostatic mechanism where we move up and down from the normal or average or healthy concentration of hormone in the blood. So understanding this diagram, we can see that the pituitary gland releases more or less TSH. The gland that is going to be affected by this from our study notes has to be the thyroid gland. And that's going to then adjust the level of thyroxine being released depending on what the body needs. So if less TSH is going to be released by the pituitary gland, that targets the thyroid gland. Remember that TSH stands for thyroid stimulating hormone. If there's less of it, there's going to be less stimulation of the thyroid. And in response to that, the level of thyroxine goes down. So the thyroid gland being less stimulated will produce less of its hormone. As that level of thyroxine goes down, the pituitary gland picks up, there's way too little thyroxine in the blood. It releases more thyroid stimulating hormone. That targets the thyroid gland and tells it, come on, wake up. And it starts to release more thyroxine. So the level of thyroxine goes up at Z. Now, after you've unpacked a diagram like this, then you go to the question. So identify the type of interaction represented by this diagram. It is a negative feedback mechanism because there's a deviation from the norm, and the negative or the opposite happens to bring it back down. Candidates quoted from the stem of the diagram and incorrectly gave the term homeostasis instead of the type of interaction represented, which is a negative feedback mechanism. Then gland Y, we've already determined is the thyroid. The disorder that is characterized by the enlargement of gland Y. So this is not stated anywhere. This is asking you to remember knowledge that has been studied. The disorder where the thyroid gland enlarges is called goiter. And then question 2 asks to state one function of thyroxine, so any one of them that you've studied. It regulates the metabolic rate, it affects the growth and functioning of the heart or even the nervous system, or it influences bone development or muscle control. Then you have to describe the role of the pituitary gland in correcting the level of thyroxine at X. Now, we've already determined that the level of thyroxine at X is going to be lower, so it's going to decrease. And it's asking you how the pituitary gland corrects this. So in other words, when the level of thyroxine is low, like it is at X, the pituitary gland is stimulated. This leads to the release of more TSH, which targets the thyroid gland, gland Y, and increases the secretion of thyroxine. Candidates read the negative feedback mechanism wrong. They had to follow the lines and arrows to understand the flow of hormones between the two glands. And it is a bit of a confusing diagram, but that's why it's important to unpack this before you actually tackle questions. Question 344, explain why continuous undersecretion of thyroxine may lead to an increase in body mass. So this relates to you understanding the functions of thyroxine in the body, in that it controls the metabolic rate. So if you have hypothyroidism, an undersecretion of thyroxine, the metabolic rate will be very low. This means that there's going to be a decreased use of nutrients like glucose. So metabolic rate refers to processes like respiration, etc., that's going to burn up fuels. So if you're not consuming that through respiration, then it's going to be stored as fat. And that's why the body mass increases in the case of someone who has hypothyroidism. The candidates had difficulty explaining and interpreting the question on how a decreased metabolic rate reduced cellular respiration, causing less glucose to be used and more of it to be stored as fat. They incorrectly referred to nutrients as food or calories. Then the last scientific investigation was conducted to determine the effect of auxins on plant tropism. The investigation was conducted by having 12 plants of the same species divided into 3 groups. You can see them on the picture, group A, B, and C. And there were 4 plants in each, even though the picture only shows us 1. All the plants were kept in darkness for 72 hours during the investigation. The growth tips of group A received no treatment. We can see that the growth tips of group B, it was cut off, replaced by a piece of agar that contains auxins. And then they even tell you that agar is a jelly-like substance through which other substances can diffuse. And then in C, the growth tips were cut off and replaced by a piece of agar, but there was no auxins present in it. So question 1 for this investigation, give the independent variable and 2 variables that were controlled. Now, to determine an independent variable, you always go to the aim of the investigation. It says, to determine the effect of auxins on plant tropism. So if we then look at the pictures, the only thing that is different between them is the presence of the growth tip and how that affected the auxin concentration. So group A will have a growth tip with auxins. Group B has auxins in jelly agar, which kind of represents the growth tip. And then C has jelly agar with no auxins in it. So it's kind of like having no growth tip at all. So what we're actually manipulating, what the independent variable here is the presence of auxins, whether they are there or not. Now, candidates incorrectly wrote the effect of the auxin. So they took that whole phrase out of the aim. And this is incorrect. We went into quite a lot of detail in this on last year's Diagnostic Report Teaching and Learning tool, which you can download from our website. If you write the effect of something for the independent variable, you are actually stating the dependent variable, because the effect of the auxins is what we are measuring. That is the dependent variable. We want to see what their effect is on plant tropism. What we manipulate is whether there is or isn't any auxin to do that. So the auxin is the independent variable. 2 variables that were controlled, you can read through the setup. They tell you that the same species was divided into 3 groups. All the plants were kept in darkness, and they were all kept there for 72 hours. So you could have selected any of those 3 controlled variables. Candidates described how the variables were controlled, in other words, dividing the species into 3 groups, keeping the plants in darkness, instead of exactly what the control variable was. They also wrote about phototropism, yet the plants were kept in darkness for 72 hours. Question 352, give the expected result of the plants in Group A and Group C. So in Group A, we have a plant stem that has had no changes to it whatsoever. You don't expect anything out of the ordinary to happen here. As this whole plant is in the darkness, the auxins that are present in its present growth tip, the growth tip is still there, cannot respond to light at all. So it's just going to grow straight upwards. Then in Group C, you don't have a growth tip and you don't have any auxins. Remembering that auxins affect tropic movements, but no phototropism is present here because there is no light. Auxins also affect cell elongation. So the fact that there is no auxins present means that there will not be any upward growth of this growth tip. It's not that no growth will occur, right? It's that no upward growth occurs, because the plant as a whole is still going to grow in other ways. It can still grow laterally, it can still develop roots, etc. It's just that because we've removed the growth tip, no upward growth will take place in group C. But what we will find is that maybe lateral branches will develop, because the growth tip no longer inhibits the lateral buds from developing. So learners could have said any one of those two things to get the mark. Question 353, how was the reliability of this investigation ensured? Going back to the typical responses, that reliability has to do with increasing the sample size and also random sampling or repeating the investigation. These are generic responses. So learners are discouraged from writing generic responses, but rather using those to formulate a response based on what was done in this investigation. So we can see in terms of repeating the investigation, they don't mention anything about that. They don't talk about anything that could hint to random sampling. So the other factor that could increase reliability is the increasing of the sampling size. So how large was the sample size? Now, yes, the investigators used 12 plants of the same species, but each group only consisted of 4 plants. So the answer for them and how they increased reliability is that each group had 4 plants in it instead of just 1 plant. So you're not comparing 3 plants, you're actually comparing 12 plants in total. And the diagnostic report says that many candidates incorrectly referred to the fact that 12 plants were used, but the 3 groups of plants were treated differently. So the sample in each group was about 4 plants big, which is more reliable than having 1 plant in each group. So therefore, it's not the total sample size that makes the investigation reliable, but the sample size of each group that received the treatment or no treatment in this case. Then question 4, after 72 hours, the stems of the plants in group B bent towards the right side. Explain these results. Now, it's important to note that the plants were all in darkness. So many candidates went off on a tangent about phototropism, which is not actually applicable in this context because there is no light for the auxins to respond to. So what candidates needed to understand is that the auxins were placed inside the agar, and you can see that it's placed off-center on the tip of the stem. Now, auxins always diffuse downwards, which means that if they diffuse directly down, there's going to be a higher concentration of auxins on the left side of the stem. As auxins contribute to cell elongation, there's going to be more growth, more cell elongation on the left side of the stem. So basically, the entire plant will bend towards the right side. The stem will grow towards the right-hand side, and that's why even in darkness, this particular stem responded as if it were exposed to light. But it wasn't. It's just about where the auxin concentration was highest based on how the agar was placed on top of the stem that was removed. So many candidates could not relate that auxins in the agar actually diffused out of it, even though it's mentioned in the introduction of the paragraph. It says agar is a jelly-like substance through which other substances can diffuse. They could identify the function of auxins and the role that it plays in growth, but not how this growth brought about the bending, the cell elongation and the bending of the stem to the right-hand side. We have a very, very nice single-page summary on phototropisms and other tropic movements in the latest Grade 12 Part 1 Life Sciences Study Guide and class text. Then lastly, suggestions for improvement on question 3. Hormones inhibit or stimulate a response in an effector or target organ. They do not perform the response themselves. So when learners talk about hormones, it's important that they emphasize inhibit or stimulate. Emphasis must be placed on the words more or less when teaching homeostasis in the endocrine system. So more ADH is released, more water is absorbed. Not just ADH is released, water is reabsorbed. ADH is always being released, but with homeostasis, more or less of it will be released at different times depending on what the body needs. So there's never a case in which there is no adrenaline in your body or no ADH in your body. We need to place more emphasis on scientific investigations and their design. Learners should be taught the value of each design element and how it contributes to a valid investigation, and we've provided a lot of revision in this regard in previous teaching and learning tools. Make sure learners know all the functions of auxins in plants, not just tropisms, but emphasize the fact that those happen as a result of its growth function. It causes growth and cell elongation. That's the only way it could respond to tropisms. Expose learners to the practical design of the various hormone experiments with auxins, as I've mentioned, there is a very nice one-page summary of all of them in our latest grade 12 Part 1 Life Sciences class text and study guide. If you found this video useful, give it a like and subscribe to our channel so you don't miss any new episodes. Follow us on Instagram or Facebook to stay on top of the latest TAS news and launches. So that's it for now from the Answer Series, your key to exam success.